will start from introducing myself. My name is Joanna Zemła. I work at the Institute of Nuclear Physics, Polish Academy of Sciences. Our affiliation may sound strange for you, but in the Institute there is a large group of people working doing interdisciplinary research, and among them there's the group of Professor Lekka, where I'm working, and Navdeep is with us for a few months so far. So during today's lecture, I will show you some examples of application of atomic force microscopy for characterization of biological samples. Ricardo's lecture was a fantastic introduction from the theoretical point of view. My lecture will be more focused on showing you examples to what type of samples can be examined with AFM, but not only, because in recent years, the development of new techniques, new approaches to characterize viscoelastic properties of biosamples is evolving. So, as it was stated in the project, so today we will have some information about rheometry, deformability cytometry, optical tweezers, or patch clamp techniques. Ricardo already showed you a bit of history of AFM. In this slide, I'm showing you the history of AFM focused on when and who and to examine what applied AFM for the first time in years. So from the early years of AFM, when it started to be used in liquid, first tapping and high-resolution images of proteins in liquid were performed. Then force spectroscopy measurements of cancer cells, which were made by Professor Lekka in 1999. Toshio Ando is an expert. He is developing high-speed AFMs for imaging proteins mainly. In 90s, the single molecule force spectroscopy and dynamic force spectroscopy methods were implemented to study receptor-ligand interactions, to learn more about the binding and unbinding of these types of molecules. And after 2000, we have single cell force spectroscopy measurements. So here the cell is the probe, and the oscillatory rheology introduced in the group of Professor Daniel Navajas and applied for AFM. So what type of samples and what type of measurements? So of course, topography, of course, force spectroscopy. So in the beginning, we were looking at the apparent Young's modulus, but now with the development, we can examine viscoelastic properties of cells, so rheological measurements. Adhesion, so single molecule, single cell interaction with many different configurations, where you have either single molecules on the substrate and cell as a probe, or vice versa. The cell and its receptors are the examined object, and the ligand is on the probe. And more complicated, more complex systems, not only single cells, but also tissues, and in recent years, three-dimensional cell cultures such as spheroids. So regardless of the experiment in AFM, we get the approach and the retract force curve, and from each part we can get information. So we can get information, we can get information about the elasticity, viscoelasticity, adhesion, but also in the case of cells, there is a part of a curve which gives us information about the properties of glycocalyx. This is the structure on the surface of the cells. This is a kind of a brush of proteins, glycans, and other, sorry, that's not the direction, and other biological stuff. So at the very first contact of the probe with the cell, it meets this glycocalyx structure, which is different for different type of cells, and in case of cancerous cells, it's more abundant. It can be thicker, which can influence the analysis of the force curves. Cellular nanomechanics by AFM has been tested to check what is the variability of the data we can get depending on the system, on the sample preparation, on the substrate. This has already been mentioned. So on the left, there is a graph showing just one scan. Here is the topography, and here is the changes in the Young's modulus along one scan of the AFM. So we can see that in cells, this local properties, the local elasticity changes a lot. It was also shown that depending on the presence of adhesive proteins, which form so-called focal adhesions, these are clusters of proteins and that are responsible for attachment of the cell to the substrate or to the extracellular matrix. And we can see here that depending whether this vinculin is present or not, we also see variation of Young's modulus. And what already is was already said today, like in general, if we look at the single cell level, in most cases, this is not also the general true. Cancerous cells are more deformable than normal cells. But in three-dimensional environment, when we have the extracellular matrix, the whole structure becomes is stiffer in case of pathological versus normal tissue. This variance, the fact that the values we obtain during the measurement depend on so many factors, it is very important to work on protocols that will allow to get consistent data. This is one of the examples of such approaches. Like 6 labs in Europe were doing the measurements of pancreatic cells, and they compared the values of the apparent Young's modulus they obtained without and with stiff bottom effect correction. So as you can see, if the cells are not high, so if the cell layer is thin, this is more, the height of the cells is less than 4 micrometers, then this stiff bottom effect is has a huge influence. After when the thickness of the cell increases, this is what already Ricardo told, then this stiff bottom effect influence of that stiff bottom effect disappears. What was also concluded in this research is that if we map elastic properties over the cell, so if we are doing mapping experiments and we want to compare distributions of data, it's better to look at medians rather than standard deviation, which gives the higher degree of agreement if we compare data obtained in different labs and also with different techniques, like here we have AFM and indenter. So this was general introduction about nanomechanics, and now let's go to viscoelasticity. This is a reminder for you. So on the right, we have a scheme of a structure of the cell, so which is a very complex matter. So we have a stiff nucleus, a lot of polymer, biopolymers inside, forming cytoskeleton, and also the cell membrane that also has its own stiffness. On the left, you can see the visualization from a side of a probe indenting actin filaments in the cell, so you can see how they bend under the load. So the standard approach of AFM measurement is focused on looking at elastic properties of cells. However, because of the complexity of the system, which also includes cytoplasm, which is inside the cell, these are viscoelastic materials. And what will be shown later, this visco part is quite important, and it gives us more information that can allow us to distinguish, to find more differences between systems. So the viscoelastic properties of cells can be modeled in various ways. So one of the popular models commonly used is a polymer model. So we imagine a cell filled with a mesh of cross-linked polymer chains. We can see this object as a poroelastic model. So we have a kind of a wet sponge that everything inside is fully viscous and it's fluid. Soft glassy model that we see a cell as a container of particles which are trapped in the local potential minimum, or already today mentioned standard linear solid model. So we have small deformations and time-dependent changes. Also, there are some models where we see a cell as a semi-permeable water balloon, and actually we only care about the membrane here in this case. So to the mechanistic, the AFM force spectroscopy measurements are actually can be rheological measurements if we observe how the cells, the object deforms in time under the force, and depending whether this is a normal force or tangential shear force, different equations are applied. But in general, we get two components, viscosity describing viscous and elastic properties. For biological materials, for low strains, the ratio between the relation between stress and strain is linear. However, if we increase the stress, if we increase the time, this behavior is non-linear. So how to obtain the viscoelastic information about the viscoelastic properties of the object? So there are three techniques. So in the first case, we impose constant strain and observe relaxation of the material. In the creep experiment, we do the opposite. So we impose the constant stress. So in case of AFM, this would be that we should keep a constant force, and then we observe deformation, changes in deformation. And the third case, already you've already seen these equations. So oscillatory rheology. So we have input signal, and we observe the response from the material, and from the difference in phase shift and the amplitude changes, we can calculate the storage and loss modulus. So storage modulus gives us information about elastic properties of the material, and loss modulus about viscous is a viscous part, so it tells us how much energy has been dissipated in the material. So in AFM system, if we have experiment and we are applying a pyramidal probe, if we impose oscillations of different frequencies at small amplitudes, then we get the response of the material from the changes of the position of the cantilever in the detector. And from applying the Snedon model, we get equations allowing us to obtain parameters describing viscoelastic properties of the material. On the right, you can see an example of one measurement on bladder cancer cell. So in black, we have storage modulus. In red, we have loss modulus. If we compare these characteristics with the graph on the left, you can see that there are that this result resembles the behavior of a polymer network, which is presented here. So at low frequencies, we observe that elastic properties are that the elastic modulus, the storage modulus is larger than loss modulus, so the cell is more solid. The behavior of the property of the polymer is more solid-like. Then we have glass transition zone when the structure freezes and the mobility of the chains is restricted, and under the load, those chains can break, or the system can remodel. So if we come back to the cell, which we can imagine as a polymer network, we can say that under certain loads, we force the rearrangement of the cytoskeleton elements, not only actin, but also intermittent filaments and tubulin. And that's why we observed that after that for high frequencies, the cells start behave like a fluid-like material. Here we apply the single power law fits. The characteristics of such response of cells in general is described with power loss, single, double. It depends on the system. Why I showed that? So coming back to the beginnings, for bladder cancer cells, it's been shown that normal have higher values of the apparent Young's modulus than cancerous cells, but we cannot distinguish between cancer cells of different grades. The same happens if we go and evaluate this relation in 3D cultures. So we cannot differentiate between cancer types. So the question is whether the viscoelastic rheological measurement will give us more information that will actually allow us to go a step further. So we examined 3 different cancer cell lines: normal, transitional, and low-grade carcinoma. And we can see that the viscoelastic, the rheological characteristics of the cell line is different. So the HCV cells have the larger storage modulus, while the low-grade cells have the smallest storage modulus. Additionally, you can see that there's a difference in transition frequency. So this is the point where we observe for which the loss and storage modulus are equal. If we present this data as a loss factor, we can see this. We can see that in case of low-grade cells, the input of the viscous properties to the overall mechanical properties of these cells is the highest, and for normal cells is the lowest. So this approach gives us possibility to distinguish not only between normal and cancerous cells, but also between cells of different grades. Again, for 3D cell cultures, we also have the same effect, that we can distinguish between different cancer cell lines. We applied these methods also to check what is the cell response to the cytoskeleton targeting drugs. Cytoskeleton targeting drugs is a new family of chemicals in recent years used to treat cancer. Some of them target, most of them which are used in clinics, they are targeting tubulin. There are also chemicals that target actin, but they are not applied for treatment because there's a lot of actin in healthy cells as well. So we just want to kill the pathological ones. So again, we studied here. We studied two cell lines, so transitional and low-grade cancer, and they were treated with Taxol, one of the microtubule targeting agent, and we observed that in time we observed changes in mechanical properties of these cells when exposed to the drug. So the low-grade cells show increasement in the apparent Young's modulus, while T24 cells they soften, and then there is just a slight change towards increased elastic modulus, and we were wondering what is happening, why that happens. So biological examinations showed that in case of transitional cancer cells, we observe increasement of vimentin, which are intermittent filaments. So there is some kind of a crosstalk between those three cytoskeletal elements that when we damage one of them, the other two reorganize to avoid changes in mechanical properties of the cells, which are important for metastasis. In case of the low-grade cancer cells, we see actually a drop in vimentin and increasement in tubulins. So the cells become stiffer because the structure of the cytoskeleton is dominated by tubulin network, and microtubules are very stiff biopolymers. Other drugs were tested on pancreatic cancer cells, and as you can see, depending on the concentration, depending on the exposition time, we observe changes in mechanical properties. This can be relevant with respect to the development of resistance of cancer cells to treatment. In the labs, we work with cells that are grown on two-dimensional substrates. However, in our system, most of the cells are for a time in a suspended state. So we were wondering how does this morphological difference translates to change to the difference in viscoelastic properties. So here we examined melanoma cancer cells. At the top line, you can see actin labeled in green and vimentin intermittent filaments labeled in red. Interesting thing is that vimentin intermittent filaments, they can reach to cell philopodia, and that this polymer structure is quite abundant. While in suspended cells, we don't see nice chains, we just see the fluorescent signal, so we know that it is there. The same is with the microtubule structure. So here microtubules are concentrated along the nuclei. Here we see the signal from all the cell interior. The cells were probed with different probe types. So we used conical and tipless probe. And for tipless, regardless of whether it was tipless or conical probe, suspended-like cells behave more viscous-like compared to the spread ones, which is quite obvious. But the question was whether is this the matter of the fact that in suspended cells, actin is concentrated around the cell membrane, forming very thick actin cortex, or do we actually also probe deeper? So we checked that by targeting vimentin intermittent filaments. So here you can see that we no longer have nicely developed intermittent filament network, and as a result, G prime, G double prime were lower, and the transition frequency shifted. So for spread cells, it shifted to over 100 Hertz, and for suspended like cells, it was very high. So this free chains of vimentin, they could remodel the system inside and react to the strains we were imposing to cells. Another approach is to apply relaxation experiments. So we indent the cell and then we wait. We can wait different times, and after a certain time, we retract. As it was mentioned today, to such behavior, such force curve, we can fit Zener model and obtain viscoelastic parameters from that measurement. So here you can see only the part of when the cell, when we were observing the relaxation of the cell, fitted with the standard linear solid model or Zener model. This is the same model, but then, with two different names. And we have three parameters. The equilibrium elastic modulus. This gives us information about elastic properties of cells at the equilibrium state. So if we impose the deformation, we wait, the cell relaxes, and at some point we get the equilibrium state. So this is the modulus describing this effect. Also, we have elastic component related relative to the relaxation process. So this is dominated by the first part of this curve. And the last one parameter is the relaxation time, which is used to obtain the so-called Deborah number. That is kind of a parameter similar to loss tangents in case of oscillation measurements, but the interpretation is the opposite. So for elastic materials, the loss tangent goes to 0. For viscous goes to 1. For Deborah number, it's opposite. So for the more elastic material, the higher the Deborah number. This approach we used to study the cells which of the shape of which was imposed by the chemical and topographical pattern. So with soft lithographic techniques, we have imprinted lectin patterns on the substrate modified with some kind of silane. And here you can see the images from the optical microscope combined with AFM, that the cells actually grow on the patterns and they change the shape. You can see that this shape is elongated. This was confirmed also with fluorescence staining of cytoskeletal components. So in green we have actin filaments, in red we have microtubules. What are lectins? Because this is important. Lectins is a group of proteins that bind specifically to sugar residues. This specificity of binding is very, very important. And in case of cancerous cells, it is also important because with cancer progression, the abundance of different sugar groups, the presence of different sugar groups is related to the cancer grade. So one could think that if we have lectin pattern and that we have cancerous cells, they should be very happy to just attach to the lectin pattern. So we only see here that they are on patterns, they are elongated, but the properties change depending on the properties of the substrate. So we have two types of controls, uncoated glass and silanized substrate, and two types of lectins patterned on the substrates. So from elastic modulus, you can see that in case of normal cells, the only significant difference is between APTES and DBA pattern. In case of low-grade cells, we have also just two pairs of differences. The same for transitional carcinoma. We have quite wide distributions of data. So we can see that substrate modification changes the phenotype, but this change is not well reflected by Young's modulus. So if we look into the rheological data and compare how the equilibrium modulus changes, you can see that this parameter is much more sensitive to the properties of cells, and the properties of the cells were modified by the contact with the substrate. This conclusion is evaluated by applying statistical analysis called Cleave's delta. These are just heat maps showing how much more likely that is that the random value from one group is larger or lower than the random value from the other group. So if we have values here close to 1, that means that those distributions are totally separated. And it says that this parameter allows us to distinguish between two cell lines, between two cancerous cell lines. This is also confirmed here, where I show you all three rows. Sorry. Rows. Not rows. Okay, on the top, you can see the parameters, distributions of the parameter for 3 cell lines on each substrate type. And what is important here is that you can see that the relation between each pair of those cell lines is not preserved. So this substrate modification changes viscoelastic phenotype of cells. So the results we can obtain on one type of substrate, they won't be translated if we put cells on a different substrate, on a different protein. This is quite relevant if we talk about automation of AFM measurements on singular cells. Depending on the approach we want to apply, whether we want to position cells geometrically on the substrate, then we have to use some kind of substrate pattern and some kind of protein to attach those cells in the areas we want them to be. And that means that we are changing their viscoelastic properties. The other approach is let them grow wherever they are and train the AFM to find by itself the cells in the optical image the system has. So depending on the approach, we have to have in mind different things. I'm sorry, I need help because it froze. Oh no, okay. Going further, AFM is not the only equipment used to evaluate viscoelastic properties of biological samples. One group of these machines are optical or magnetic tweezers. The idea, let's say, it's quite similar. So we use either electric or magnetic field to trap the molecule. We put inside for example, inside the cell, either a molecule, either a microsphere with different refractive index or some magnetic particle, and we observe by changing the fields. We observe the movement of the particle inside the cell, and from that we can obtain the information about viscoelastic properties, because usually we move the particles, making them to oscillate. So this is some kind of oscillation rheological measurement. What are the limitations? Sorry, it's not working. For optical tweezers, we have a laser beam. So this is this can be a disadvantage because we have a high power laser, and that can hit the cell. And we know that many types of cells are sensitive to light. So the exposure to light starts processes of cell death. So this is one of the drawbacks. In case of magnetic tweezers, we don't have that problem. We can access different changes of properties of different objects, not only cells but also proteins. We can also rotate, manipulate. There's a lot of options here. There is just one example of a particle placed in a three-dimensional polymer network that is oscillating, and the changes in the phase and amplitude give us information about the properties of the material. Sorry, yeah, this is DNA. So this is a particle inside a DNA. This can be surprising, I think, for you, that rheometers, which are microscale apparatus, they are commonly used to measure viscosity and viscoelasticity or other rheological properties of all kinds of materials, starting from food, from cosmetics, to asphalt, cement, like different types. And in recent years, it started to be used for biological samples. 10 years ago, it was shown that with rheometer, that's if for you, if you don't know, you have a scheme. So in general, we have two parallel plates. The fact parallel is very important in this measurement, and the top plate can be rotated or oscillated over the second plate. The sample is in between them, and the sample has to fill all the space between the plates. It cannot go outside. It cannot be. We cannot have any. It has to be filled. It has to be filled. So with the single cells, suspended cells, the problem was to control the parallel, the fact that the plates are parallel here. Here, the bottom plate is a glass plate that allows us to observe the sample from, observe the sample from the bottom. So here you can see labeled suspended cells, and they are in the culture medium, and we perform the amplitude sweep measurement. So we oscillate with the constant frequency, but we change the formation. So we increase amplitude of the formation in time. And as you can see, for a gap of 5 micrometers, we obtain data where we can see linear behavior of storage modulus with increasing amplitude. So we characterize so-called linear viscoelastic range. So in theory, all the measurements should be performed within this linear viscoelastic range. These are results for a different cell line because the cell's height was larger. We had to increase the gap size had to be increased, and just to show for the control, if we measure just the medium, we can see that we can prove that we actually sense the presence of the cells. Other methods allowing to calculate, to check viscoelastic properties of cells are high throughput methods. So here we use microfluidic channels or deformation cytometry. So here we also have a channel. These are PDMS channels. So here you have an example when we were observing the transition time of different types of cancerous cells, and that could be related as well to the width of the channel and height of the channel is a little bit smaller than the average size of the cell, and we are checking the diameter and how wide the cells become in the channel. And from that, we can get information also about not only transition time, which is it results from the viscoelastic properties of the cells, but also about the viscoelastic properties of the cells. Here you can see another example. Oh, sorry, the movie is not working. So here is another example of cells in the channel. So the cells, we have a pressure pump that is pumping cells through the channel. Across the channel, we have a different pressure of the liquid in which the cells are suspended, and that causes the changes in the shape of the cell. And from that, also from the changes in the shape of the cell, and also from, because when they go through the channel, they also rotate. From that rotation, we can also get information about viscoelastic properties. So this is the summary. So storage, loss modulus, and stiffness. Now I will show you a few examples of three-dimensional systems. So here we are examining viscoelastic properties first on the alginate beads to validate the new method. So the new approach was to apply the fiber. We are attaching it to the surface of the spheroid, and in the system which is connected to the tube and the pump, we are creating under pressure in the system that which we are controlling in time. We are changing that in time. We are oscillating with that. And from information how much the part of the cell was sucked into the fiber and what is the oscillation time, you can see the change in time, so this oscillatory signal. Not going into details, the equations, they just show that the properties depend on the pipette radius, actually on the length of the sample that is in the capillary, and the difference of the pressure. And here we have a comparison overlap of AFM data in yellow and hydraulic force spectroscopy data in blue. And what we can see here is that they are in a good agreement, but hydraulic force spectroscopy gives us possibility to check the properties of the material at lower frequency range, which is not possible to obtain with AFM system. This result shows behavior on different cell spheroids. So you can see the top images show you AFM data. Here we have HFS data. And if you can recall the graph from a few previous slides, we can actually observe the same behavior as for polymers. So at very low frequencies, the system behaves more fluid-like, then solid-like, and again, fluid-like. To fit this data, so for AFM data, we were fitting double power law, while for HFS data, this model had to be modified by adding one of the dashpots. Another approach is quite simple idea, but it was that simple that probably was hard to come up with that. So people from Glasgow, they invented so-called rheoptical assay. So you are just not working. Just putting cell spheroid between glass slides. So more details will be here. And this is just the image of the idea, but this spheroid is suspended in PBS, so it's not just like dry spheroid. And from the bottom, with the phase contrast microscope, we observe how the spheroid changes the shape under the load. So by observing how the shape of the spheroid's cross-section changes in time, using this quite easy equations, we can see evolution of strain and stress in time, and from that we get storage and loss modulus. And here we can see that the data we get are in a good agreement. So for direct measurement we have lines, and the points are modeled. So there is a good agreement in between. We had examples of measurements on cells. We have examples measurements on 3D cell spheroids. And now I wanted to show you some results obtained from tissues. So this is a part of collaboration with University of Lausanne. People are working on a new approach to cure Duchenne muscular dystrophy. We got samples from a mouse model. This mouse were injected with it's called mesoangioblast cells. This is a sort of primary cells, and there were different numbers of this transplantations. And if we used AFM to study that, we see that for healthy, which is B6 seed, the tissue is stiffer, while the dystrophic muscle has lower elastic modulus values. If we compare those distributions with distributions of the elastic modulus obtained for transplanted muscles, we can see that at nanoscale for single transplantation, there is no change in the elastic Young's modulus, while after 3 transplantations, we can see 2 peaks in the distribution. So we have bimodal distribution. So that means that there are areas where the transplanted cells did not do the job, and the area where they actually improved they reconstructed the connections between cells and improved the structure of the muscle. The same muscle samples were measured with rheometer. So we are looking what happens at macro scale. And at macro scale, we can see that regardless of implantation time, we see overall improvement of the muscle elastic properties, because compared to diseased, which are in magenta, everything is within error bars at the same level. However, if we look at if we compare the histopathology of the tissue, we see that there is a difference, that still, even after transplantation, we have some scar tissue, some collagen in between. So this muscle did not recover in 100%. And this is what actually is reflected in loss tangent. Because if we compare the loss tangent values for those 4 groups, we see that, of course, healthy behave more solid-like, while diseased are more fluid-like, and the transplanted samples are in between. So the structure, the inner structure, the connections between the cells and the extracellular matrix in treated samples improved, but not in 100%. At this point, I would like to finish the part about viscoelastic properties of cells. If you have any questions, we can ask now, or we can continue. Yeah. Yeah, this is a very important factor because the best approach would be measure the fresh sample, like directly from the biopsy. This is technically difficult because the equipment have to wait and then data analysis. So we were evaluating that. We tested 3 different protocols. And from the tested protocols, deep freezing of the tissue is the best one because it helps to keep the properties of the tissue. And this is our paper from 2018. These were asthma samples taken from patients. So we are comparing samples taken directly from the patients and also treated with the protocol. And deep freezing is a good approach to preserve the properties of the tissue. So, well, single cells, you have to, you can use you can use paraformaldehyde at low concentration. This is important, at low concentration and short time. You should find a paper by Targosz Korecka. I don't remember the year. She was showing what is the difference in the values you get from measuring the Young's modulus with AFM from the same not treated with anything, live cells, and those with which were treated with small concentration of paraformaldehyde. As far as I recall, there was no difference. Yeah. It's a reversed Hertz model. So we keep it as a infinite, and the cell is the bead, like. So if we have in rheometer, if we have parallel plates geometry, you can vary the width from few micrometers to nanometers. The only limitation is actually more you less control the data. You can have more less reliable data if you apply a very small diameter of the plates. And this relation between the gap, the diameter, it also has to be checked versus the sample you are probing. If you have very large particles, for example, if you're measuring suspension and you have very large microparticles, then from the size of the microparticles, you have to validate. It's the same as with probe resolution. So there is some limit for let's say, rheometer measurement resolution. In rheometer, you can also have a second geometry where you have a cone plate over the parallel plate, and here you cannot control the gap. The gap is controlled by the angle of the cone. But this gives you, in plane plate geometry, you have gradient of deformation. So the largest deformations are at the edge of the plate. If you have a cone plate geometry, then this is compensated. So you have the same deformation over all over the sample. Yep. Yeah, of course. This is This is actually a discussion I had in the beginning with Professor Lekka, because she started working with like this traditional force spectroscopy modes, and she prefers high resolution mapping of the cells. With oscillations, you cannot do that, because I always say that, like, imagine there is someone holding a pencil and is stabbing your arm constantly, like rather faster than later, you will react. So this is the same. Also, there are some types of cells that are very difficult, not only to measure, but also to do even high-speed imaging. We had some collaboration. People came with the glioma cells, and you couldn't do the topography image because you can, like, in real time observe the cell was like moving, like the probe was touching, and she was like, no, I don't want that, I'm going away. So yeah, this is a thing that we have to observe. Yeah, it is also important. Yeah, depending on the live cell, whether it is like a mitotic state, whether it's dividing or dying, these properties change. That's why there are two approaches. One of the approach is to study synchronized cells. So you can have a population of cells that is at the same stage of life. The other approach is we don't care about that. We just do a lot of measurements and to see the distribution. But what type of measurement you mean? Yeah, and we are checking with AFM, for example. So this here, it depends on indentation depth and the probe geometry, as Ricardo said. Everything depends on the probe geometry. Because also, if you have the conical probe, the information you get is like more like in Z axis. So everything near the probe and more beneath. If you have a sphere, this information is more from going X, Y rather than Z. And for oscillations, to avoid the bottom effect, you use small indentation depths, typically around 1 micrometer. So even if you have a cell that, I don't know, has some particles in it, unless this particle is directly below your probe, it doesn't affect. Okay. Actually, I thought that I would be faster, but okay. I will start about adhesion. This will be, as I checked, during next meeting, you will have also lecture focusing about single molecule for spectroscopy. So I will just go quite a little bit faster through those slides, just to show you the concept. So as I already mentioned, the cell has a lot of proteins sticking out of the membrane, and they form the so-called glycocalyx. And in that glycocalyx, we have integrins and syndicants. These are molecules which are mediating in cell-cell adhesion, cell ECM adhesion, generally whatever the cell wants to touch, it uses these receptors. And from different type of imaging techniques, you can see that this is not a structure that we can just ignore. So at the single molecule level, we have protein and ligand, and when they bind through different type of interactions. So we can have ionic bonds, hydrogen bonds, hydrophobic van der Waals interactions. And besides the beside that, also geometry of the molecule is important. So it's kind of that it has to fit, like a key to the door, it has to fit. Sometimes it is possible that the molecules will bind that if they don't exactly fit, then they form less stable complexes, as you can see on the right, and this is the presentation of that. So to study such interactions, this binding, unbinding of these complexes, we have to work in native environment on isolated complexes. We have to study many molecules, use high force resolution, and do a lot of experiments. That's the truth. So already mentioned that we can have different configurations, receptors on the substrate, ligands on the probe, receptor on the cell, ligands on the cell, also cell as a probe. And later I will show you that also other new improvements have been shown in recent years. The crucial element for this experiment is probe modification. So to attach the ligand to the probe, there were established two types protocols. So the first protocol was using glutáraldehyde. So we have AFM probe covered with silane aptes. Then we attach glutáraldehyde, and then we attach the ligand. This has limitation. It works, but has limitation that the connection between glutáraldehyde and the ligand is stiff. So this ligand cannot move. So if the binding site is somewhere on the other side, this won't work. So that's why in group of Hinterdorfer, they established new protocol. So we use PEG ligands. And depending on what type of protein we want to study, we use shorter or longer PEG ligands. This is also good because the length of the PEG ligands allows us to avoid false results coming from non-specific adhesion, which can also happen. If we have the ligand, then we actually shift all the unbinding events a little bit on further distances on the force curve. For this measurement, it's very important the properties of the cantilever. They are very crucial. So the cantilever has to be soft, and the shorter the better. Because long cantilevers, this is already Ricardo mentioned, they interact with molecules of the medium you are measuring in. So if we have long cantilever, many molecules, we have a lot of noise. The shorter cantilever, soft cantilever, the sensitivity increases. So to say more about the force and the energy that is needed to unbind, to rupture the complex, we perform these measurements with various loading rates. Loading rate is proportional to pulling speed. So for each complex, you have to measure a set of experiments at different pulling speeds to get information about the energy landscape of the complex. So the loading rate is actually defined in this part of the force curve, and the rupture force is the force counted, like the maximum force in this case from the baseline. So the other remark, which not everyone remembers, is that data obtained for single molecule force spectroscopy without control experiment are no data. You have to block the interaction and repeat the measurements to be sure that your observation is actually this complex binding and not other stuff, that this is actually this specific interaction. Usually you do that by adding, for example, if you have lectins which bind sugars, then you add sugars to the solution, wait a little bit and start scanning later on, and you should observe like that the number of force curves with any events, unbinding events will drop significantly. I mentioned earlier syndicans and integrins because these are the groups of adhesive proteins that were studied by other people, but in my lab. And the question was, which complex unbinds first? Because those two form together a complex binding responsible for adhesion of cells to extracellular matrix. So we are probing syndicans and integrins with, for example, with antibody to syndicans to obtain the information about the energy hallmark. So there are three models that are fitted to the data to obtain the description how this separation occurs. So we have free energy barrier, and depending on the model, we can get information where this barrier is and how high, what is the height, so how much energy we need to give to the system to rupture the complex. The basic approach is Bell-Evans model. So here, the limitation is that we assume constant position of the barrier. So the position of the barrier is constant here, which of course is a huge approximation to the real life. This model has been developed further, and Dudko-Hammer-Szabo DHS model. So here they added that part that gives you the information about the change in the position of the energy barrier. And the third model is a Friedel model that encounters one more thing that happens during the experiment. At low loading rates, that means low pulling speeds, we can observe so-called rebinding. So we have a complex, we rupture it, but the distance between the molecules is still very close, and it is energetically convenient for them, they bind again. So in this model, that is included. So that gives us the true information about the shape, about the landscape, energy landscape of this complex. So for syndicans, you can see that the shape of the barrier is different depending on the syndican type. And here, if we compare syndicans with integrins, we see integrins are in purple, that they rupture faster than syndicans. So the detachment of cells starts from detachment of integrins and then syndicans. This I will skip. Now the inverted system. So now we are probing the proteins on the substrate, and the cell is our probe. So this approach has several factors we have to be aware of. First of all, the contact area. We can obtain like the average cell size, but it is really hard to define the contact area with adhesion with single cell spectroscopy experiments. This large contact area results in very complicated, and this one is complicated, but not as complicated as it can be, retraction force curves, where you can have jumps and tethers. So these are so in time, complex by complex, they begin to rupture until we see the final rupture of the last complex between the cell and the substrate. So this is just example for different extracellular matrix proteins. We see that depending on the protein, the characteristics of the curves is different. So we have one step for control, but for fibronectin and vitronectin, we have 6 and 9 steps. Steps are actually the tethers here. And this is example how people like to complicate life more. So a group of Professor Alessandro Podesta in Milano, they used extracellular matrix. So they used extracellular matrix from tissue. So they had tissue slices. They removed the cells. Only the extracellular matrix stayed. Then with laser, they cut out squares of that tissue. They put that on the probe, and they were checking the adhesion between this ECM slice and cells. And here is a complicated force curve for analysis. So they were comparing, checking all the tethers, all the jumps, and all other stuff. And by combining the results of the single cell for spectroscopy and staining the parts of ECM they used on presence of different ECM proteins, collagen 4, laminin, beta 1, fibronectin. They correlated the results and show the difference depending on from which area of the ECM they took the probing part, then it influenced the results. So the last thing I wanted to show you, this is scanning of this is single molecule for spectroscopy performed on bacteria trails. So bacteria at the first contact with the substrate, they start twitching, they produce their extracellular matrix, and this protein is deposited behind them. So this is the optical view from atomic force microscope. These are trails labeled proteins behind the green bacteria cells. And here you can see the images collected with single molecule force spectroscopy of height of bacteria cells, stiffness, this is relative, and adhesion. And what you can see that adhesion, in adhesion, you see broader trails of bacteria. This is a result that the white pyramid probe was used. So even the probe was next to the trail, it still could bind proteins from with molecules which are on the side of the pyramid. But what is interesting, here you can also get a whole gallery of different type of force curves. Without AI, analyzing such data is quite time-consuming. It's extremely time-consuming because, as you can see, they are very complex, of different shapes, and yeah, this is a place where also some automatization could be applied. And this is my last slide, showing you the newest AFM modification called Fluid FM. So these are actually kind of modules attached to AFMs that already people have. So the idea here is that we have a probe with a channel inside, and we can use that probe either to inject cells with something or to suck the cell to the probe and avoid the probe functionalization and just speed up the single cell for spectroscopy measurements by using this equipment. We have it in our lab for a few months, and well, it's hard to say right now whether it's faster or not. We'll see in time. Okay, and this is the end for today. So these are people I'm working with, of course, thanks to all the people in the consortium, and thank you for your attention. Sorry, I had to speed up the second part. I don't know if you have any questions. No. You're free. If you don't have, unless you have questions. Yeah, people are doing that. Yeah, they are putting DNA on the substrate, and they approach with the probe and try to detach that or unfold it. Yeah, different types. No, for DNA are rather optical tweezers.